Good morning, everyone, and welcome to our inaugural Virtual Research Café 10.0. It's actually nice to see a lot of familiar faces in the Zoom picture here on my monitor. This is a new pilot program that we're starting here as part of STEMnet. So it's great to see all of you here. I'd like to first thank Monica Alacon, who did our admin analyst for STEMnet, for doing a lot of the logistics on the front end and for publicizing the event. The goal of the cafe is to help foster research collaborations across the CSU and to catalyze the potential submission of joint proposals. The cafe brings together recently hired CSU faculty to share their work in a very relaxed setting. And each of them will have 10 minutes. Note the reason for the 10.0 here. We hope you'll find opportunities for collaboration as well as learn about a new area of research that may impact your own program. As I mentioned, I'm Frank Gomez, the ED of STEMnet. Feel free to contact any presenter in the chat box. I will be looking through them and bringing those questions to the forefront. But do not hesitate when the presenters are not speaking to ask them questions before or after their presentations if need be. At the end of the presentations, everybody will come together at the end for Q&A, and that will go as long as questions come to the forefront, okay? Our first speaker is Alex Klotz from the Department of Physics at Cal State Long Beach. So Alex, the floor is yours.
Hold on, the sharing is just getting going here. All right, so thank you for putting this together and all of you for attending. As mentioned, I'm Alex Klotz. I'm a new professor in the physics department at Cal State Long Beach. I've been here about a year now. So before I tell you what I do, I'll tell you a bit about who I am. So I'm from Canada originally, grew up in Toronto. I did my undergrad at Queen's University, which is a few hours away, in physics, and then grad school at McGill in Montreal, also in physics. I defended my PhD in 2015 and then moved to the United States, where I worked as a postdoc at MIT in the chemical engineering department for a few years before moving to California almost a year ago today and starting at CSU Long Beach. So most of my work is in the field of experimental biophysics, but I was also briefly famous for a more accurate calculation of how long it would take to fall through a tunnel through the center of the Earth, which was a worldwide news item for about 2 weeks a few years ago. There's a picture of when I was on the Canadian Discovery Channel. And my latest hobby has been road biking. It's nice that it never rains or snows in Southern California. It's pretty robust against COVID-induced disruptions. So what do I do is the next question. And the title said something about big DNA molecules. So there's sort of a stylized rendering of a DNA molecule. And I think when a lot of people picture DNA or what DNA does or what it's like, it's something like this. There's a sequence corresponding to the chemical structure, which has some biological relevance, a gene that codes for a protein or something like that. That's not how I look at DNA. To me, DNA is more like this. It's just essentially a microscopic spaghetti noodle. So I'm from a field that just strips away all biological relevance from DNA and uses it as a model system to study the physics of materials. And the type of material exemplified by DNA is a polymer. Polymers are these long, stringy macromolecules that make up a lot of modern materials, especially soft materials. And I don't think it's too controversial to say that by learning more about the physics of molecules, we can have a better understanding of the physics of materials. But it's very difficult to study synthetic polymers one molecule at a time. But it was realized that DNA is basically a very big, naturally occurring polymer. And the technology used to study it for biomedical reasons got good enough to study it for physics as well. So there's a lot we can learn about the physics of polymers by looking at genomic length DNA molecules. There are actual biological reasons that to understand the physics of genomic length DNA molecules. There are a few modern sequencing technologies that operate by manipulating genomic length molecules. These include nanopore sequencing, nanochannel mapping, and zero-mode waveguide sequencing. And to enable these technologies, it's necessary to understand polymer physics as it applies to DNA. For example, how a gigantic molecule fits through a tiny pore. That's the kind of physics question that has to be answered to move, in this case, nanopore sequencing forward to a viable technology. The human genome, if you stretch the molecules out entirely, reaches about 4 meters. We have about 100 trillion cells. How long is 400 trillion meters? That is long enough to stretch to the sun 600 times. It's about a light month. So how do you take so much material and package it into our tiny cell nuclei and still have the molecule useful and accessible and unentangled? That's a polymer physics question, just not one that I have an answer to. So that's some motivation as to why we study big DNA molecules. Share a bit about what I actually do. So a project I was working on as a postdoc looked at the physics of knots in DNA. So anything that is long and stringy and subject to randomness is mathematically guaranteed to form knots. You may be familiar with the macroscopic version that I have pictured here. But DNA also forms knots, including our biological DNA, which we have proteins to untie. So I did a series of experiments using fluorescence optical microscopy and microfluidics to entangle and stretch individual DNA molecules. You can see a video from one of my experiments here of a big knot untying. So the behavior of polymer systems is strongly dependent on the amount of entanglement between the different molecules. So knotted DNA is a way of understanding the effects of entanglement on a single molecule level. We can also understand how knots move around molecules such that we can either control them or prevent them. For example, get rid of unwanted knots that are messing up with genetic sequencing devices, clogging nanopores or whatever. So I like this video from one of my experiments. That's a 10 micron scale bar, just showing a molecule. It starts with a very big knot. The knot gets to the end of the molecule, partially unties. We now have a medium-sized knot. That knot does its own thing, eventually partially unties into a smaller knot, and then completely unties. And you can see the molecule stretching a bit more after each stage of untying. So that's the type of thing we investigated with these experiments and also with a lot of computer simulations. Lately, especially since starting my new lab, I've been interested in these DNA structures called kinetoblasts. So this is the parasite in the top left that causes African sleeping sickness. And they have very weird mitochondrial DNA. Instead of one loop of DNA like ours, they have about 5,000 loops that are all linked together like medieval chainmail armor. You can see some close-up pictures of those loops here. And these are some fluorescence images from my experiments where they look like these 5-micron wide fluorescent jellyfish. So I've been investigating these for about 2 years, and it's one of the things that I wanted to start looking at in my lab, except my lab got shut down for 4 months immediately after I opened it. So we reopened last week, and this is currently the thing we're looking at. So these experiments with kinetoblasts are fun to do and fun to watch. There's some of my favorite movies below. But they also have exotic material properties. So for example, we can ask, how does a material respond to external stresses if the molecules aren't held together by chemical bonds, but by these mechanical linkages? And kinetoblasts are effectively two-dimensional. There's a lot of interest in 2D materials, but all the experiments basically have to be done at absolute zero in perfect vacuums. But these exist in like room temperature, noisy, wet environments. So it's a way of trying to sort of research that can get 2D materials out of the lab by seeing how they interact under out of lab conditions. So, like I said, there were other things that I had hoped to investigate, but I had a bit of a roadblock with the global pandemic. In terms of collaborations, so if you define one axis that runs from mathematics to biology, and then another axis that runs from pure to applied research, I'm sort of directly in the middle of both those axes. So happy to collaborate with people who are more applied or less applied, or wetter or drier compared to biophysics. So if there are people who do simulations of molecules or other types of modeling who want to compare their simulations to a real physical system, I'm interested in that. If there are biologists or life scientists who have an in vivo system of choice that could use a more controlled in vitro investigation, also interested in that. And I'm sure there are engineers out there who want to know if something is physically possible before they start designing a technology, and I'm happy with that as well. But if anyone is interested enough and friendly enough, I'm sure we have the creativity to find some sort of common ground. So I've had two projects funded to work with some other CSU investigators, which I hope to actually start doing soon. One is with Veronica Jimenez Ortiz at CSU Fullerton. So she studies these parasites that have the weird kinetoplast DNA, and I'm hoping that we can look at how the loops are connected using nanopore sequencing to diagnose parasite infections. And with Ryan Blair at Long Beach Mathematics, I'm basically having students just destroy kinetoplasts either with laser beams or with enzymes or something. And we're using that destructive testing to understand sort of mathematically how these links fit together. So that's about it for this presentation. Thank you for listening. And maybe next time I do this, you'll see your face up there as well. Thank you.
Great, thank you, Alex, for a very interesting short seminar, if we can call it seminar. So our next speaker is Matin Pirouz from Fresno State. She's in the Department of Computer Science. So Matin, who seems to be on a vacation someplace in the tropics, its floor is yours.
Thank you, Frank, for the introduction. Let me share my slides. So, as Frank introduced me, I'm in Vegas, Nevada. In computer science, we usually APT, and this is my third year. As for the hobbies, recently I've been reading books related to my field, and I wanted to read for a long time. Well, I could not remember the to a vacation this summer to see friends, and didn't allow me. So my vacation changed to a staycation. So as for today, I have prepared one of the reasons. I'm going to talk about some of my research pillars first. Are in big data, complex networks, and people ask me, so what's the difference? Basically, a complex data that didn't exist could be the size of data that is big, or it could be that makes it complex, or it can be the accuracy of data. When we have so, or it can be speed of highest.
So Matin, it seems you were breaking up pretty badly. Do you want to, do you hear us? Are you able to hear me now? You're on mute right now.
Can you hear me?
Now I can hear you. You want to give it one more try to see, once you went with your slides, you broke up badly. So let's try it one more time, okay? Because you seem to be okay now. Let's give it one more try, okay? So share your screen, please.
So how much of the talk have you been able to hear?
You would need to start
Frank?
from the beginning.
Okay. Hello, everyone. I'm Matin Pirouz. I'm sorry for the technology problem. So I finished my PhD in University of Las Vegas in computer science. Usually we don't do postdocs. So I came here as an ABD, and then this is my third year as an assistant professor. Are you still able to hear me?
Yes, I can hear you. You're a little jumpy in terms of the video, but we can hear you still.
Let me, oh, that's share a screen. Okay, that's not possible either. Okay, I try to continue. Please let me know each time the voice is not clear so that I can maybe change something.
Okay.
Matin, maybe try to turn down your volume a little bit.
Sure, let me see. Does this work?
You sound a little bit clearer. Go ahead.
Okay. So research pillars. There are three main research interests for me: big data, complex networks, and data mining. In terms of big data, many times I get the question of what is big data? What's the difference between big data and just data? Why do you have to separate it? Well, the answer to that is, with normal data, a farmer here in Central Valley would be able to ask his nephew or son or daughter to put the data on an Excel sheet and just handle it that way. But with big data, that's not possible. Excel sheet won't be able to process this data. And this is just in terms of volume. So if your data is getting bigger than a certain size, the traditional technology is not able to handle it. But there are also other factors like speed. If the speed of producing data, generating that data is too fast, then the traditional technology wouldn't be able to handle it. Also, in terms of fault in data, if we know there is fault in our data, then we need another complex system to be able to handle that data. And these days, we know with technology, with mobile phone, with games, and with the new technology introduced, we have different types of data in the single format or in the single file. And that makes it difficult for traditional methods to process that data. So these are the factors that make big data and make it different from the regular size data that we had before. The second research interest for me is complex networks. Again, what's the difference between networks and complex networks? When a network is changing dynamically and constantly, that makes it a complex network because we cannot use static models to calculate the features of that network. And the third main point is data mining, which is the process of discovering patterns, meaningful patterns from large data. So these are the main interests of mine and what I have been working on the past few years. Now, the project I wanted to talk to you about today is a project we have started with some colleagues in some other CSUs and also Fresno State. It's called Trilemma problem. As you can see here, we have a triangle. We have health, food, and ecosystem. So if we look at any of these components, if we look, for example, at the food, we know there is still hunger in the world, and we have problem of not having enough food. So what if we expand our farming and we have more food now so that we can feed everybody? That will hurt our ecosystem because more farming means more changing to ecosystem. But it helps health of human being so that we can have less hunger. On the other hand, if we think about ecosystem and we want to have it healthy, then probably we need to have less farming or a different type of farming. Also, health follows the same problem. They are all interconnected. Each one of them changes. The other one also changes. So the project we went after is to explore and design and develop a system that can have this trilemma, this triangle in it, and can play with different variables. And to find a prediction system that can actually predict and tell us what happens if we change something in the food system, in the food component. How much does it change the health of human being? How much does it change the ecosystem? So talking about trilemma and working with it, we kind of expanded the trilemma because we figured it's missing a component. The component here in the middle, human behavior and socioeconomics, we realized if we remove this, these three are not going to change by themselves. So what is changing this balance is human behavior, how we farm, how much we farm, how our health is under compromise because of not having enough food or because of the ecosystem that is going to global warming and other pollutions. So now this new component will give us a handle on how to control and how to affect other components of this trilemma. So development of prediction of framework, which is a graph model to estimate the changes brought to trilemma due to human activity, like smart farming, automation-like changes. All of these are changes that humans are bringing to our ecosystem and our trilemma. First question is, what are the interconnected control variables and their impact on the trilemma? So if we look at each component, what we see is a multi-layer network, which is also temporal and has different variables inside. For example, when we talk about food system, you can grow your own vegetables in your yard, especially in Central Valley, we can do that. Or you can go to a farmer's market and buy your vegetables. Or you can go to a supermarket. Or you can go to some farm and ask them to pick by yourself. So there are different methods to do the same, to have the same result at the end. So when we are calculating components for only food component here, there are variables that should be considered with different factors because not everybody farm in their own backyard, right? Not everybody goes to farmer's market. The majority of people shop at the supermarket, but still we need to consider all the variables involved. So when we are talking about such a thing, we are talking about different layers of network. At the end, they give you the final destination and the final result, which is the same, but the path you are taking toward them is different. So the path which is different are the edges in there, and they make the difference for you to how to reach there. And these variables make the difference in equation in our calculation. This is only one component. Then if we want to talk about intra-edges between this component, what is the relationship between food and health? So how are these two connected? what variables are working there, and how do we factor these variables into our calculation. That's the next point for us. So question 2, now that we know about the problem, we know how to approach the problem, we have built a system that is connected inside each component and between the different components, and is giving us measurements, calculation, numbers that we can talk about, we can have graphs, we can give advice based on. But how to measure and work each component of the Trilemma, in fact, how to know if we are right, how to have metrics for it, where do they work? So that's the next component of our research, which comes after building the system, to know, okay, where do we stay and how do we optimize the system that we already have. So who is on our team and what skills they are bringing? First of all, it's me. I am there to do the data manipulation, transformation, visualization, big data modeling and data analytics, graph theory. Technically, everything related to data would be connected to me to handle. And I think one important component that I have to talk about is the data itself. So in this project, we are working with different data, from agriculture big data to weather, to socioeconomics data about human, to health data. So all of this data are coming from different organizations, and we are gathering this data and working on crunching these numbers and making them to a format that is both understandable to the team, and we can feed it to the system so that the system can make decisions based on that. The second member of our team is Nina Robson from CSU Fullerton. And she is in charge of modeling and designing the automation for the system. So one thing to add about that is this automation, we decided in order to case study and to know our measurements, we need to build systems to make automation some kind of product. How the automation will change the farming, the food and ecosystem triangle as a case for us. Next is Dr. Yu Yang from CSU Long Beach. He is in charge of optimization and small scale modeling. Next, we have Dr. Erwin Balderrama from Maths Department, Fresno State. He is our statistician, data integration from heterogeneous sources and Bayesian interpretation from big data. Next, we have Dr. Shahab Tayyeb. He is our electrical engineering. He's in charge of data capture, sensors in the farms, and sensors for the environment, weather, data transfer, Internet of Things, and network science. Physical network, not complex network. Next addition to our team is Dr. Rajir Riyar. He's in charge of farming automation and data veracity. What we are looking for, who we are looking for to complete our team is an agroecologist, socioeconomist, and many more. So if you find yourself interested in the project, if you see yourself somewhere in the project that can help, please go ahead and contact me. Here is my email address. We would love to have your contribution and collaboration in our team. Thank you for listening, and Frank, the floor is yours.
Thank you, Matin, and sorry we had some technical difficulties in the beginning, but thank you for an interesting talk. So
it was probably on my side, so I'm sorry, but was it okay for the rest of the time?
Yes, it worked fine. So great. So last but not least is Stevan Pecic from Cal State Fullerton in the Department of Chemistry and Biochemistry. So Stevan, your turn. Good luck.
Okay, thank you. I hope you see my screen. Okay, so yes, I would like first to thank organizers for inviting me. It's really great being here. I would like actually that next time we actually do this face-to-face. So today I'm going to talk about one of the projects that's going on in my lab. We are working on development of inhibitors of two enzymes. That's why it's called the multitarget. And we are typical medicinal chemistry lab, so we can do design, we do some organic synthesis. We also equipped to do in vitro screening. We can actually do kinetics and measure inhibition against these two enzymes. And we also have some limited skills to perform molecular modeling studies. We mostly do predictions of pharmacokinetics in order to design new drugs. So we are aiming to inhibit two enzymes, and both of these enzymes are involved in pain and also inflammation. So these compounds are possible therapeutics for pain and chronic inflammation. So because of the limited time here, I'm not going to talk about current options in pain management, but we know there are opioid drugs, and there are also non-steroidal anti-inflammatory drugs. We have completely novel approach. We want first to inhibit enzyme that's called the soluble epsilon hydrolase. This is an enzyme that is part of arachidonic acid metabolism. I will show you this in next slide. And the main substrate for this enzyme is called epoxy-icosahexaenoic acid. These acids are converted by this enzyme into biologically inactive dihydroxy-icosahexaenoic acids. So here you can see the arachidonic acid metabolic pathway. So arachidonic acid in our body is not, it's usually bound to plasma membrane. And then when you have inflammation or pain, it's released in blood, and it's been degraded by different enzymes. A group of enzymes called the cyclooxygenase, they convert arachidonic acid into prostaglandins and prostacyclins. These are all mediators of pain and inflammation. And actually, there are many drugs, FDA-approved drugs, that are inhibiting these enzymes, and they are being used for pain and inflammation, like aspirin, ibuprofen, also many other, as I said, non-inflammatory non-steroidal anti-inflammatory drugs, NSAIDs. The next group of enzymes that are metabolizing arachidonic acid is called lipoxygenase enzymes. These enzymes are most located in lungs, and they also degrade arachidonic acid into leukotrienes, and they are all involved in pathogenesis of asthma, allergies, and other pulmonary diseases. And there are FDA-approved drugs actually that are inhibiting these enzymes and are beneficial for treatment of asthma and different lung diseases. The third pathway that we are interested in is being for a while in research, like last 20-30 years. So it's been explored, but there is still no available drugs on market that are actually inhibiting any of the enzymes in this pathway. So as I mentioned in my first slide, these acids, epoxy-icosahexaenoic acids, they are beneficial. They have anti-inflammatory effects. They also have anti-hypertensive effect. So these are really good acids to be around in our body. And there is an enzyme, as I mentioned, that is degrading these acids into this metabolite, which is biologically inactive. So if we inhibit this enzyme, we will actually be able to increase the level of these acids, and that can be beneficial for many diseases, but we are looking something for pain and inflammation. There are many ways to elevate the levels of ETs, so you can actually have agonists or anything else, but in my lab we are actually looking to find inhibitors of this enzyme. There are many drugs, as I said, because this research is going around for the last 20-30 years. The first generation of these enzymes, of these inhibitors, are mostly derivatives of, they're looking like substrate. They are still quite potent. You can see the low nanomolar range, they can inhibit the enzyme. But the second generation looks more like drugs, because this is what in medicinal chemistry we don't consider as like drug-like compounds, but second generation is more drug-like, and they're still quite potent. And as you can see, they all share common structural features, which we call urea moiety. In my lab, we are actually looking for compounds that are non-urea based, and they are still like SEH inhibitors. The other enzyme that we're looking to inhibit is called fatty acid amide hydrolase. That's the abbreviation FA. We have in our brain two types of receptors, CB1 and CB2. These are cannabinoid receptors, and these are G protein kappa receptors. They've been activated by three classes of compounds that we classify as cannabinoids. There are phytocannabinoids, synthetic cannabinoids, and endocannabinoids. Phytocannabinoids, they are mostly derived from marijuana. They are like from plant derived. This is like example THC. We also have synthetic cannabinoids, like this guy here. They are mostly synthesized in the labs. And we are interested more in endocannabinoids. These are cannabinoids that are produced by our body in our liver, and then when we have pain or inflammation, these cannabinoids will bind to CB1 and CB2 receptors, and they will have pain alleviation effect. So as I mentioned, there are much benefit having these guys in our body. They produce pain alleviation. They also have anti-inflammatory effects. They also have some other effects for some other diseases in our body. They have a lot of applications, and what is really good with these endocannabinoids, because they're made by our body. They specifically recognize these receptors, so there are no off-target binding, so there are no side effects like you have with THC or synthetic cannabinoids, so there is no side effects. But unfortunately, there is also, like our biochemistry system works, we also have an enzyme present in our body that degrades these endocannabinoids and convert them into arachidonic acid and ethanolamine. And this is inactive in terms of pain and inflammation. So this pathway also is well known. There are many FA inhibitors on market. Just based on the names here, you can see that Big Pharma is quite interested in these compounds, like Pfizer, Johnson & Johnson, Bristol Myers Squibb. So many pharmaceutical compounds. We cannot compete with them. So I'm not looking for new FA inhibitors. What we are kind of working on is to combining these two pathways to inhibit these two enzymes, because they are connected, as you can see, via this arachidonic acid system. And then if we can inhibit simultaneously this enzyme and this enzyme, we will have elevated levels of both EETs and anandamides, and that will basically have a beneficial effect in pain management. So what is the rationale for this study? In 2015, we had a paper where people had tested separately SEH inhibitor and FA inhibitor in several rodent models of pain. So there were rats that were treated with separately SEH and FA inhibitor, and this is isobolographic curve. This is a theoretical efficacy here, and this is actually experimental value. And everything below this isobolographic curve we consider as a synergism, which means that giving SEH inhibitors and FA inhibitor together, they will actually have a much better efficacy on treating pain. Now, so we have this, somebody will ask why we just don't develop SEH and FA and just give this mixture together. Why we are actually looking to find only one compound that can inhibit in the same time both enzymes. So the answer to that, there are many benefits. First of all, if you have one compound that is safe to be taken, that doesn't mean it's safe independently. It doesn't mean it's safe if you take it in combination. There are many different drug-drug interactions. Even if we are looking at some simple safe drugs like, I don't know, Advil or aspirin, we don't know how they will react if you start taking them together. There is also, FDA doesn't like mixture of drugs because of this. It's also difficult to develop these drugs, to predict their doses, pharmacodynamics, pharmacokinetics. So it's very complex story. Also patient compliance, patients like also take one drug comparing to 2 or 3. And in medicinal chemistry, we call this approach DML, which stands for Design Multiple Ligands. So we want to find one drug that will simultaneously inhibit 2 or more targets. In our case, it's 2 enzymes. We call it polypharmacology, and that's what we're doing in my lab. So this is the general flow of things that we do in my lab. So as medicinal chemist, we work on this first part. As I said, we do design, we do synthesis, we do biological evaluation. We are looking for collaboration is to move this project to the next phase, where we want to test compounds for their stability, predict some of their toxicity, and also some pharmacokinetics. This is what I'm talking about in terms of pharmacokinetics, absorption, distribution, metabolism, excretion, if they can also cross blood-brain barrier. We want to develop these compounds for preclinical model, test them in mice and rats, and then move them into clinical studies. So far, we were successful, and we found actually many compounds that have dual inhibition, which we consider if they can inhibit enzyme below 50 nanomolar against both enzymes. And we had a few of these compounds. We still continue working on this scheme, but we are looking for any collaboration in this area here. For this project, I've been actually awarded also a grant from NIH. And I would also like to thank my department, BioChemistry department in Cal State Fullerton. I've been there since fall 2018. On this project, I also collaborate with Hammock group from UC Davis. They were pioneers in discovering SEH and the inhibition of this enzyme. And finally, I would like to acknowledge all my students working in the lab. You can read more about our projects and about my students and me, and research publications at website. I have this, it's quite catchy. So please visit and also reach out if you want to collaborate. Again, I would like to thank you for your time, and Frank and everybody else who organized this. And I hope I see you next year, not virtual, like face-to-face meeting. Thank you so much.
Great, thanks, Stevan. And I appreciate everybody's presentations. If the presenters can please come on. And for anybody in the audience that would like to ask questions, if you can, please turn your video on when you do that. But the floor is now open for everybody out there. If you have questions, you don't have to go through me. So just, you're on.
I've got a question for Alex. Good morning, Alex.
Hi.
This may not be quite in your domain, but it does definitely have to do with knots in DNA. Is it known that there are any human conditions or diseases or situations in which DNA knots and that causes a problem?
Not really. The closest thing is that there are diseases associated with the failure of topoisomerase, which unties knots, but you can't make the connection that the knots are causing the disease.
Okay, thank you.
I guess one other thing is that in cancerous cells, the mitochondrial DNA forms catenanes, kind of like the kinetoplasts.
So Stevan, I'm wearing my non-admin hat now at the CO, but back to my days of being a real scientist, a chemist. What makes adamantyl moieties so so often used as inhibitors to enzymes?
Yeah, that's an excellent question. I really found in many different projects I found adamantyl moiety present in drugs. It's highly hydrophobic and it's also bulky, but it's not aromatic, which is also good for some parts of the receptors. So most receptors that we are targeting for drug discovery are G protein coupled receptors. They usually have this kind of in binding pocket. There is usually a part that is highly hydrophobic and doesn't tolerate sometimes aromatic rings, but still needs some hydrophobicity there. So this part of the drug is really, you have a lot of carbons there, and they're again bulky, but they're also compact. So it's not just a chain. So you don't have much of the rotation there, which is also good. So this is quite rigid structure, but also in the same time hydrophobic. And I found it in many drugs present. But unfortunately, it's also, I noticed it's not very metabolically stable. So our liver enzymes quite easily recognize this part and break it down, which is not good. I found these compounds to have stability of few minutes to top 1 hour in our body, which is something not good for drug discovery. You don't want to take drug every 5 minutes, right? So.
Any other questions for our presenters out there, or any other questions in regards to life in Cowstid LA in general?
I did have a private question that Bio Joe sent me, who also asked one live, but it was about why these kinetoplasts are made of connected loops, and basically it's because their DNA is encrypted and they have this gene editing scheme to edit their own RNA, and they have to keep all the loops together. So if anybody was wondering, that's why.
Okay, well, if we don't have any more questions, I certainly like to thank our presenters today for giving us a little summer research teaser regarding what they're doing. And you can always contact the presenters at Long Beach, Fresno, or Fullerton, or if you don't remember their names or where they're at, you can always contact the STEMNet's office, where we can provide you their logistics. And we'll be having another one on September the 9th with 3 more speakers. And I see some of those speakers attended today. And I hope everybody stays safe and healthy in our new world that we're all experiencing here in the CSU system. And I certainly appreciate all of you participating today. And hopefully we'll meet as Stevan said, in person one day. So everybody, have a great day. Bye.
Thank you. Thank you all. Bye.